Hey guys, welcome back. So this question is cool because it ties in two things you've learned about. It ties in what you know about secreted proteins. So remember from midterm 1, what does every secreted protein have? Right, it's going to have a signal peptide. And hopefully it reminds you of recently during the cell bio part where we were learning about the endomembrane system. So this is the endomembrane system. Right, so all of these organelles are part of it. And they're all made of the same membrane material, so that's why one can pinch off. So here we see something like rough ER pinching off and fusing with the Golgi. That's super allowed to happen. They're fluid. And we want to put all of these events in order, but we don't want to put any events that secreted protein would not go through. So let's look at some that we might be able to cross out. So for example, obviously only one of these can be true. They're the same thing. Where does transcription happen? Right, transcription or going from DNA to RNA happens where the DNA is, so it happens in the nucleus. So I'm going to cross this guy out. Hopefully that one was a freebie. Let's also compare translation. So it's possible for proteins to be translated at the rough ER, and it's also possible for proteins to be translated at the cytoplasm. The difference is, so for the signal peptide, has to travel through this entire endomembrane system. Right, so we're going to be using this arrow to help us later. And the cytoplasm isn't really part of that, right? It's going to the protein will begin its life at the rough ER and then travel through the endomembrane system. So all secreted proteins will be translated at the rough ER. They will not be translated at the cytoplasm. So we can erase those two. Right. Some proteins are translated at the cytoplasm, but those will not be secreted proteins. Cool. So let's go ahead and go through this question together, starting with what's for sure at the beginning. So before you have a protein, you have to have RNA. That step is transcription, right? So transcription in the nucleus, this happens first. I guess I'll just number it like this. Transcription in the nucleus has to happen first. After transcription, what's going to happen? Translation. Right, this is nothing new. This is where looking at the diagram comes in. So we already knew transcription and translation, that's nothing new. Now that we're at the ER area, you travel from here to the outside of the cell. So the next step is the Golgi. Before you hit the Golgi though, you need to be processed in the rough ER. So proteins are processed inside of the rough ER. Then from the rough ER, we travel to the Golgi on the cis side. So cis, you'll learn this when you get a little bit deeper to Chem 1C. Cis means same. It's the cis side of the Golgi because it's on the same side as the ER. Then it leaves the Golgi through the trans side. Trans means opposite, so it's on the opposite side as the ER. Another way to remember is it goes from C to T or in alphabetical order. Regardless, after the ER, that's when it hits the Golgi. And then you can tell it's leaving the Golgis in this little membrane sac. These are vesicles. Just a little sac of membrane that can contain things. So this would be 5. And then that vesicle will fuse with the plasma membrane, and then whatever was in here, inside of that vesicle, it just gets dumped into the outside, so completely dumped out there. So this would be last. Okay, cool. So hopefully this makes sense to you guys. If this does not make sense, come get help. And let's move on to the next one. So we are considering the chloroplast. The chloroplast is super sick. We will talk about it in the next 2 questions. So remember when we learned about our metabolism, so we went from glycolysis all the way to pyruvate oxidation. With the chloroplast, you only need to worry about 2 reactions, the light reactions and the Calvin cycle. So to no surprise that light reactions use light, and what do they use light to make? Right, they use light to make energy. In what form? Right, they make ATP, they use light to make ATP. And they also make an electron carrier. Do they make NADH? No. What do they make? They make NADPH. So you've already seen how tricky these tests can be. So definitely be, when you're studying, like be on the lookout for that, right? What if there's an answer choice, but there's NADH instead of NADPH? I remember it because I just pretend the P stands for plants, right? Then we have the Calvin cycle, and that's going to use all of the ATP and NADPH. So the light reaction makes all this energy, and the energy is used for the Calvin cycle, which uses carbon dioxide to produce glucose. So I remember the Calvin cycle, the C, it's all about carbon. So carbon dioxide and glucose. Cool. The light reaction is all about light to make energy in these two forms. We're going to go a little bit more in depth on that in the next question. Cool. So comparing the mitochondria and the chloroplast, it's so cool because they're opposites in so many ways. So if the metabolism part was good for you, like you remember what's happening in the mitochondria, everything that happens in the chloroplast is the opposite. So let's go through these together. And let's just go through the mitochondria first, since we already know what happens here. So what's the carbon-based reactant that each organism consumes? So us, we start with glucose, right? Glucose doesn't go into the mitochondria, you know, it's like technically pyruvate, but whatever. The main point is we take up glucose, and what do we produce? What do we breathe out that's carbon-based? We breathe out carbon dioxide, so that's our reactant. So when you go from C6H12O6 to CO2, is that being oxidized or is that being reduced? It starts with 12 hydrogens and then it loses them, so this gets oxidized. And think about the role of H2 and O2. So remember, that was in our electron transport chain. Do you guys remember the role of oxygen in the electron transport chain? Hopefully you remember we breathe in oxygen, so it can be our terminal electron acceptor. This is why we breathe in oxygen. I think you can remember that we breathe in oxygen and we produce the water. Cool. I'll save D for the end. Now we can go ahead and do the chloroplast. What do plants take in? Right, think about like, why do people plant trees? Why do they say trees are good for the environment? Because they take up carbon dioxide, so that's a reactant. And what do they turn that carbon dioxide into in the Calvin cycle? They turn it into sugar, which is so sick. So they're doing the opposite of what we're doing. We're taking sugar and breathing out carbon dioxide. They're taking in gaseous carbon dioxide and putting out sugar. Cool. When you go from carbon dioxide to this, is it being oxidized or is it being reduced? It's gaining hydrogen, so it's getting reduced. Reduction involves gain. If the reduction oxidation part was hard for you on the midterm, by the way, go ahead and check out my previous video about oxidation reduction. Hopefully that'll clear it up. And next, think about water and O2. Think about another reason why are plants so good. Not only do they take up carbon dioxide, but they also produce oxygen. And they do the opposite of what we do. So they take water and then they turn that into oxygen. So they're oxidizing water. Cool. So before we go into this next part, we kind of need to talk about these organelles, right? So the mitochondria, each of them have a double membrane, right? One membrane here, one membrane here. Each have a double membrane. Here we have the inner membrane space in between the two membranes, and then we have the matrix in the middle. The chloroplast is a little different. You have these stacks of things. The stacks are called grana. And just each individual grana, let me write that here. In one grana, there are multiple thylakoids. So both of these organelles are similar and they have a little pocket. So like the pocket of the mitochondria is the matrix. And the pocket of the chloroplast is all of these thylakoids. All right, so do you remember in our mitochondria, where does all of the H plus accumulate? So both of these electron transport change. chains with chemiosmosis and H+, in us, remember the H+ is in intermembrane space. So it's kind of like in the outer compartment. So cool. So in chloroplast, it's the opposite. So the H+ is not in the outer compartment. It's inside of the little pocket. So the H+ is in the opposite place too. It's inside of the thylakoid. Right, so here, the H+ is in the intermembrane space. Right, kind of in the outer area. In the chloroplast, we have H+ in the thylakoid, or the more interior space. Okay, cool. So I hope that you feel like excited about how opposite the chloroplast and the mitochondria are. And I will see you guys next week with some more cell bio.